Hello, my name is Nicole, and today I will be presenting on my work in germline genomic biomarkers of grade reclassification of low-risk prostate cancer at prostatectomy. So the main clinical problem in prostate cancer, let me just zoom in on my background here, so the main clinical problem is accurate risk stratification to inform a proper management strategy. The risk presented by the cancer is primarily determined by a pathologist grading a biopsied sample of the prostate. Unfortunately, it is not uncommon for the biopsied sample to not accurately reflect the composition of the entire tumor, leading to misclassification. A misclassification can be detected if a man's prostate is removed during surgery and the whole specimen is pathologically evaluated. A biomarker that could predict a grade reclassification at prostatectomy would aid in management decision-making prior to prostatectomy. Now, since genetic features are predictive of the diagnosis of prostate cancer, for example, the BRCA2 gene and polygenic risk scores for prostate cancer diagnosis, there is potential for genetics to also be predictive of prostate cancer outcomes like upgrading. To test this, I use the upgrading reference set, which is an EDRN cohort established for the validation of upgrading biomarkers. It consists of men diagnosed with grade group 1 prostate cancer who underwent radical prostatectomy and were assessed for a change in their grade group in their resected prostate. Controls have no upgrading, and cases have upgrading to grade group 2 or above. Now, researchers are blind to the case control status of this reference set and can submit putative biomarkers for validation to the EDRN. My primary objectives were to first characterize the germline genetic features of this cohort, then develop a genetic biomarker of upgrading, and finally validate it with the EDRN. So to accomplish this, let me scroll over to my method schematic. To accomplish this, a subset of the upgrading reference set, or the URS, was selected for whole genome sequencing. After bioinformatics processing, I performed ancestry calling, annotation for pathogenic variants in the known prostate cancer risk gene, BRCA2, and the calculation of 2 known polygenic risk scores for prostate cancer diagnosis. I combined BRCA2 carrier status and the 2 polygenic risk scores into 2 genetic risk of upgrading scores using a decision tree, which is shown down here. The resulting 2 PRS-based biomarkers were then sent to EDRN for validation. Moving on to my results. This section displays the results of the genetic characterization. So first, in this figure, we have pathogenic or likely pathogenic variants, which were annotated in these 7 genes, with 3 carriers of the MUTH variant and 1 carrier of a BRCA2 variant. I compared these carrier frequencies to an external cohort of men with intermediate risk prostate cancer and found both of the MUTH and BRCA2 carrier frequencies to be lower in the upgrading reference set, though these proportions were not statistically significant. Next, we have the 2 principal components of ancestry. With the URS samples being plotted over the 1,000 Genomes Project reference population. It is somewhat clear in the next figure on the right, but the majority of the cohort, around 80%, was of European ancestry, followed by about 12% of being African ancestry, and these results were highly concordant with self-reported race statistics. And in the final figure, I have plotted the 2 polygenic risk scores for prostate cancer diagnosis colored by ancestry. And you can see that the 2 scores were highly correlated, which was expected. And as I mentioned earlier, these 2 scores form the basis of 2 scores for the genetic risk of upgrading. And a decision tree was used to adjust the score for the single detected BRCA2 carrier to be the maximum possible polygenic risk. And then these 2 scores were then submitted to the EDRN for validation as predictors of upgrading. Moving to the second set of results, these 2 figures here show the results of the validation analysis at the EDRN. These are the receiver operator curves. And as you can see, neither score was shown to be predictive of upgrading, with areas under the curve close to 0.5. So in conclusion, this validation study in a relatively small cohort with just a single BRCA2 carrier showed that a genetic risk of upgrading biomarker that was based on BRCA2 carrier status and 2 polygenic risk scores of prostate cancer diagnosis, this biomarker was not predictive of upgrading at prostatectomy. However, the genetic data generated from the upgrading reference set remains a resource that other researchers may use to further explore the genetics of low-risk prostate cancer. Thank you very much for listening.